Okay, so DNA polymerase is actually not shown in this diagram, but I'm going to label it for you and we're going to draw a DNA polymerase in here. So here you can see that this is the original DNA molecule or the parental DNA molecule that's still intact. And you can see that DNA helicase has done its job here because the DNA double helix has been unwound or untwisted, and the bonds between the bases have been added. Now, if you look closely, you can see the red strand here represents the newly added nucleotides. So I'm going to go ahead and draw a DNA polymerase enzyme here, and we're gonna call this DNA polymerase and say that this was the enzyme responsible for adding those new nucleotides. Now that would have also happened on the original complementary strand. Okay, so I'm going to go ahead and also draw our DNA polymerase here, a little easier with a little bit thicker pen. Okay, so we have like our sloppy DNA polymerase and our little bit neater DNA polymerase. Okay, so we have DNA polymerase. Is adding new nucleotides of DNA. And that's all based on complementary base pairing. Now, here, it's not showing what nucleotides are in either the parental strand or the newly added nucleotides, but we can assume that they are complementary base pairs. You'll see more detail on that in a minute. Okay, this is a nice diagram that kind of combines what we've just talked about. You see the complementary base pairing here at the top of the molecule, and then you see the complementary base pairing here that was used to make the two new strands of DNA. Now, if you follow this purple color right here that's labeled old strand, that's part of the original or parental DNA, and you can see that on the other side too. And then in the lighter pink color, that's the new strand or the newly added nucleotides that were added using the original DNA as a template to direct the synthesis. And this diagram actually does a really nice job because right here you can actually see a new DNA nucleotide being added, a thymine being added to an adenine. But obviously DNA helicase and DNA polymerase are not shown in this diagram, but the result of their actions are shown. Okay, so the result of DNA replication is two identical strands of DNA. They are identical to the original DNA strand, and they are identical to each other. So let's look here at, this is our original or parental DNA. And it's shown in blue. Now, it's showing the result of DNA replication of this molecule here, and you can see that in the two new strands, so these are the two new double helixes of DNA that were formed. Let's see, how should we write that? Let's write it up here. You can see it in each of those two new new double helixes of DNA that half of the strand is from the parent DNA, and then The other half is new nucleotides that were added. And if you look closely, you can see that each of these two strands are identical to each other. We have CG, oh sorry, sorry, GC, CG, TA. Oh, it's the same here, GC, CG, TA. And if we look back to the original strand, GC, CG, TA. So the complementary base pairing maintains the all the DNA that was copied. It maintains it. They're all identical. Now, the word we use to describe the fact that the newly formed DNA is made up of half of the parent DNA and half new DNA is we say that DNA replication is semi-conservative. Now, you can think of semi as meaning half, like a semester is half of a school year, or semi-lunar, half of a moon. And conservative comes from to conserve, or that also means to save. So when we say that DNA replication is semi-conservative, you can think of it as in each of the two new strands that are formed, half is conserved from the parent and half is newly formed DNA. Okay, so in summary, DNA replication is based on complementary base pairing. DNA replication involves two enzymes, helicase and polymerase. Helicase unwinds and unzips or breaks the hydrogen bonds of the DNA, and polymerase adds new nucleotides to the existing DNA. This diagram does a nice overview of what's happening, of the actually it doesn't really show the complementary base pairing, but it shows the semi-conservative nature of DNA, and that each of the two new DNA molecules that are formed, half is from the parent DNA and half is newly added. Please go back and review any parts of this lesson that you did not get your notes complete for. Please let me know if you have any questions, and I hope this was helpful. Thank you. Bye.